Okay, I wanted to deal with messenger RNA kind of on its own because this is most of what we talk about when we talk about RNA processing. So messenger RNA, after it's produced, before it can leave the nucleus, has to be spliced, capped, okay, and this is a methyl group and some other goodies added to the 5' end of the messenger RNA, and then the addition of a poly A tail at the 3' end. Now it turns out that in eukaryotic cells, messenger RNA goes from like a big pre-messenger RNA to one small functional unit. And that typically happens through splicing. And so it turns out between the 60s and the 70s, people figured out that there are segments of DNA that do not equate to the messenger RNA that leaves the nucleus. And so they called this process of altering the messenger RNA splicing based on how segments were removed. So it turns out that the coding sequences of a gene are called exons because these are the fragments that exit the nucleus. The intervening sequences, the stuff, the junk, right, those are called introns. And so what happens is that you start with this long piece of messenger RNA that has segments of exons and introns in it. Okay, so I'll have exon 1 and exon 2. The intron itself gets excised out, and exon 1 and 2 get spliced, kind of a different word for glued together, all right? And then that would be your mature messenger RNA with the cap and the poly A tail at the end. So introns are removed and they stay in the nucleus. Exons exit the nucleus, and this is called RNA splicing. Now, I was goofing around here earlier, and I had to do some editing, so let me get rid of all these notes here. Now, there are 3 mechanisms that explain RNA modification. Group 1, Group 2 intron splicing, and then the spliceosome. And we're going to talk mostly about the spliceosome. And in all cases, the intron is removed. Once that little intronic piece of DNA is removed, it remains in the nucleus and it's degraded into nucleic acids where they're recycled. The cell is really good at recycling things. And so, in this way, RNA actually behaves as an enzyme. We call that a ribozyme. So remember, 99% of enzymes are proteins. 1% are made out of RNA. And not necessarily ribosomal RNA, but RNA in general. The group 1 and group 2 introns are a little different, and like I said, we're not going to spend much time really talking about those. However, here is a little example showing you that group 1 and group 2 introns use different nucleic acid derivatives to help remove segments of the intron, or to actually remove the intron. So the nucleotide actually joins the 2 exons together. In spliceosomes, we have something different going on here. Again, I was playing with this before. The spliceosome occurs, or in the spliceosome mechanism, the splicing itself takes place between branch points, all right? Let's come over here. These are called intron-exon boundaries, and the intron and the exon hold the pre-mRNA in the correct configuration so that the 2 exons can be stuck together. This is accomplished using a set of proteins called SNRs, or SN, small nuclear RNAs. So, as I already kind of identified here earlier on when I was playing around with this, there are 3 regions where you'll see segments that will interact with the SNRs. So we have, let me come back here, our intron here. Remember, this intron is coding DNA, or non-coding, sorry. It's getting late here. And then the exon is going to be like exon 1 and exon number 2. These are going to be spliced together. We have the splice site, the branch site, and the 3' splice sites, the 5' and the 3'. Pardon me. So what we have here is a situation where, let me get rid of my former notes, where exon number 1 and exon number 2 need to be glued together. Different SNRPs are going to bind to different segments of our branch points. So, SNRP U1, SNRP U2. U1 binds at the 5' branch point. U2 binds at the well, this is the branching site actually. This is the 5' splice site. And then U4, 6, and 5 bind to the 3' splice site. When this happens, this lariat formation occurs, and exonuclease activity can occur. So remember, exonuclease means that you're cutting nucleic acids. They act like an enzyme here. These two exons are glued together, and now you have I guess if this protein only had 2 exons, and some of them do, you would have a mature messenger RNA as long as you have the cap and the poly A tail on the 3' end. So why is splicing a cool thing? It turns out that the same gene can give rise to more than one protein if you don't count all the exons. What am I talking about? Oh, of course I don't have a picture of that. So let's say you have a gene with exons 1, 2, and 3. Now let me get a different color ink here, or a different color. This will be Intron 1 and Intron 2. If the cell or the enzymes stick together 1, 2, and 3, you'll have a mature RNA that contains segments 1, 2, and 3, or exons 1, 2, and 3. Okay, but now let's say this whole region here gets spliced. In other words, you lose exon number 2. Then you're going to have exons 1 and 3 put together, and that's going to give you a different protein because you're missing that intervening information. So this gives you the opportunity to generate different protein sequences based on different messenger RNA sequences that go all the way back to this alternate form of splicing. And believe it or not, this happens more often than you would think. This allows an organism to carry fewer genes in its genome. It lightens up the genome. All right. Capping, I've talked a little bit about that. This is where you'll have methylguanosine added to the 5' end, and this helps give polarity to the messenger RNA. And it usually occurs using an RNA polymerase, and the messenger RNA needs to be at least 25 bases long. And so this is what the methylguanosine looks like. Poly A tailing, and so here are just more details here. Poly A tailing occurs when an enzymatic process continually adds more A's to the end of your sequence. So you have this polyadenylation signal here within the messenger RNA. That, at the end of that, the exonuclease activity will basically start adding lots of A's to the end. And this gives a 3' polarity to your mature messenger RNA. So in addition to that, there are other things that can be added to a new strand of RNA. You can basically edit the nucleotides that are there. I'm not going to ask you guys to know about that for the exam, so don't worry about it. I just need you guys to understand the process of transcription: initiation, elongation, termination, and then RNA modification, capping, splicing, and polyadenylation. All right, folks, I hope you have a fun time watching the videos. I'll be posting those right behind these videos, but they're going to be in a separate cluster. They're imported videos from YouTube. Thanks.